Okay, so since it is already 3 o'clock, we will get started. So, welcome everyone to the last week 8 of this TA session for the course Bioengineering and Interface with Biology and Medicine. The course code is NOC 25 BT05, and once again, I just decode the pointer. Yeah, the course code is NOC 25 BT05, and my name is Arjun, and I am one of the TAs for this course for the iteration of this course from January to March 2025. So after 7 weeks, we have come to the last week of this TA session, week 8. So what I plan for this TA session is something similar to the previous 7 weeks. So we are going to go over a brief recap of whatever happened in the week 8's lectures. After that, we will be going through some sample problems for we'll be going through some sample problems for week 8, and after that we'll be solving the assignment questions that you got for week 7 and week 8, thereby completing thereby completing this discussion of all the sample problems and all the assignment questions. So without further ado, we'll just go into the week 8's lectures. So in week 8 is a continuation of week 7's lectures, and it's going to be a termination of all the course content that you have observed. So in week 7 you saw concepts which related to amino acids and proteins, and the interactions of proteins with each other, protein-protein interactions, and we also saw more about proteomics and how to study the protein-protein interaction through proteomic based approaches. And there we saw different techniques, gene sequencing techniques, and different label-free detection techniques. And then we also proceeded to see more about techniques to study protein and proteome through the interaction of two-dimensional electrophoresis as well as your DIGE, that is differential gel electrophoresis. And after that, towards the end of the week 7's lectures, we saw more into some bioinformatics tools to study this proteins. Week 8 is a continuation of your week 7's lectures, where we continued to see more techniques about to study protein and proteome, where we started about where the course instructor Dr. Sanjeeva Srivastava started about explaining the principle of mass spectrometry. So mass spectrometry is a tool which is used to analyze your if you have any sample and if you have a mix of proteins, your mass spectrometry is going to give you information about the mass of the protein, more likely the charge to mass ratio of the protein. And by keeping the charge constant, we are going to learn more about the mass of the protein. And the mass spec this is the simple schematic of how a mass spectrometer works. So basically, in your mass spectrometer, there will be a sample injection point, and upon a certain point, your sample will get ionized into ions, and your ions will be accelerated towards your field, either an electrical field or a magnetic field. And depending on the charge to mass ratio, it will go reach the detector, and the detector will process it and give you certain peaks in the mass spectrum. And depending on and the final mass spectrum, which gives you the certain peaks, the molecular ion peak corresponds to the protein, corresponds to your protein of your interest. So in the lectures, we saw two main components of a mass spectrometry system. So one system was your ionization system. So in your ionization method, you saw two techniques especially, named electrospray ionization and MALDI. Electrospray ionization, or ESI, follows a method in which your samples are present in a tube, and then a voltage gradient is applied across the ejection tube as well as your collector tube. So there is a high voltage gradient here, and there is a small nozzle here to eject your samples. So your samples eventually form a very small, fine droplet-based charged ions, which then are accelerated at a very fast rate to reach the collector. This is what your electrospray ionization method is, and this is a form of liquid ionization. In MALDI, we saw more about a solid ionization technique. So here MALDI stands for matrix assisted laser desorption ionization. So what happens is your sample is mixed with a your sample is mixed with a polymeric organic compound there are two to three we saw two to three polymers of interest which can be suitable for to be used as matrices. What happens here is the sample is mixed with your matrix, and then what happens is a highly focused laser beam is struck to the matrix, causing the generation of this ionization. So what happens is eventually the matrix coats itself around the sample and it causes disruption of the sample, and this forms the and following the process called dissolution and ionization, it results in the generation of your ions of your interest, and the resulting ions of interest are proceeded to go to the mass analyzers. So mass analyzers are instruments which are used to separate your ions based on their charge to mass ratio, and we saw two separate mass analyzers, namely time of flight, which is called as TOF, and quadrupole mass analyzer. So in TOF, what happens is here you have your sample, and the sample is accelerated once again. There is a certain space here, and this can either be an electrical field or a magnetic field or a vacuum region. The sample is made to accelerate through this region towards the detector, and based on your charge to mass ratio, uh, your charge to mass ratio which reaches the detector, it will uh, it will be detected. Just give me a second. Sorry, there was a slight disturbance at the door. But anyways, whatever. What? Once again. Sorry. Yeah. So what I was saying is, so your time of flight depends on ions which are ions which are heavier in nature, which have a higher charge to mass ratio, take longer time to travel to the detector, and ions which are like smaller in charge to mass ratio, which are very lighter, they take less time to travel to the detector. So your time of flight basically separates your um, ions based on the charge to mass ratio depending on the time that they take to reach the detector. And in case of quadrupole, what happens is you have four rods like this from the um, ion ejector to the detector. In between them, you have your four rods here, and between two rods there is a constant radio frequency field, and between the other two rods there is a constant electrical field. So it is a so what happens here is there is a presence of a dynamic electrical field, which is corresponding to our any one of the charge to mass ratio of our interest. So your quadrupole is used to selectively pass through ions which have a particular charge to mass ratio. So let's just say our ion of interest has 1600 gram per mole or 1600 daltons if we are going to talk about proteins. So let's just say our protein as a mass of our set of amino acids or peptides have a molecular mass of 1600 daltons. So the quadrupole's dynamic electrical field will be adjusted to such a extent that your ions which are only selective to 1600 daltons are allowed to pass through the detector, and ions which are like 1400 or 1500 daltons are made to strike the quadrupole rods present here. So in this way, your ions are selectively made to pass through this quadrupole rods through the detector. And after this, the detector will count the ions and it will give you an output based on the charge to mass ratio. And we also saw that in the recent times there are a lot of fusion technologies uh, which use more than one of the mass analyzers, which in some mass spectrometry we can see two mass analyzers lined up one after the other, two quadrupoles, or in more advanced systems we are seeing more than one presence of more than one mass analyzer, like systems like QTOF, which stands for quadrupole time of flight. Have a two mass analyzers where one is a quadrupole and one is a time of flight. So this helps in more efficient uh, detection of your proteins of your interest. And we also saw about other advanced mass spectrometry techniques like using eye track technology or orbit track technology or orbit track fusion technology. And And after the mass spectrometry, in the next lectures, we saw more about your protein interactions and microarrays. So in protein interactions and microarrays, the course instructor, he started to say about conventional techniques to conventional low throughput techniques to study protein and protein interactions, namely your immunoprecipitation, where you break your protein of interest into small chunks and you administer your antibody of your interest, which is specific to your target protein of interest, which causes the precipitation of the protein. And after that, you can take your protein and you can check the presence of the protein using other semi-qualitative techniques like Western blot. And we also saw more about the yeast to hybrid method. And after discussing some of the conventional techniques, we moved on to discuss about the high throughput techniques, of which predominantly it was we were discussing about protein based microarrays. So protein based microarrays are very high throughput techniques because they use very small amount of your sample, and they are also used to analyze or visualize the protein of interest in multiple samples of interest, more than 100 or 500 amounts of samples of interest. They can use through you can detect your protein of interest through your microarrays. So in microarrays, we saw two different types of microarrays. One is your abundance based microarrays, and one is your functional based microarrays. In abundance based microarrays, we saw more about your antibody or target protein arrays, where we saw more direct labeling of your protein, and we saw more about a technique called NAPA, which stands for Nucleic Acid Programmable Protein Array. And following that, we also saw more about your function based protein microarrays, where we saw techniques such as multiple spotting techniques, halo tag arrays, etc. Where depending on the protein function and depending on your capture antibodies function, it will be used to detect your this thing. And we also saw more about DNA microarrays towards the end of the lecture, where we saw their applications to study between interactions and modifications between protein and DNA. And also we saw their applications of DNA microarrays to autoantibody screening. And in the next lecture, which continued towards the protein interaction and systems biology, we saw more about your interactomics. And interactomics is the study of interaction between different protein molecules, and interactome is the sum total of all the protein interactions that are happening. And we also saw more about the importance of protein interactions and how the disruption of these protein interactions will lead to disease conditions in our body. And then we further went into look about more label-free techniques such as plasma resonance. And here is a general schematic of your surface plasma resonance, where there's a gold film, and upon a gold film there is a linker, and upon your your ligand of your interest is conjugated to your linker, and what happens is, to the gold film An incident light is passed through the gold film and at a angle called as the angle which causes the total internal reflection. Your plasmons in this gold thin film start to excite and they start to emit a energy which is being tracked through this intensity of this position. And once your analyte binds to your ligand, this causes a change in the reflectance angle of this gold thin film. Therefore, it results in a reduction in the intensity. So these kind of changes in your analyte ligand through your analyte ligand interactions are used to study your uh, certain phenomena such as protein-protein interactions, binding kinetics, and it's used to find several parameters such as your binding constant, your dissociation constant, and your association constant. And towards the end of the lecture, we also saw more about how SPR, your surface plasmon resonance, is coupled with several other techniques such as microarrays or with mass spectrometry based techniques to come at more efficient methods to calculate or to predict interactions between different molecules. And towards the end of the lecture, we also saw more about your systems biology based aspect where the course instructor started explaining about what systems biology is. So, systems biology is just a general holistic approach to study uh, study your interactions between different types of molecules on a very large scale. So systems biology is of two types. One is your model-based systems biology, and one is your data-based systems biology. In your model-based systems biology, you fundamentally try to model a system to study more about the system without disturbing it. And in your database system, you collect data from an existing system and you try to find any sorts of loopholes or unknown gaps in the system. And following that, we had another recap of bioinformatics following from our bioinformatics 1 recap. So in last week's recap, in week 7th bioinformatics, we saw how to use your NCBI database and how after entering NCBI ID, you also can get any information about your gene or protein of your interest. And then after the after exploring the NCBA database, we also saw more about BLAST and cluster omega to study more about your pairwise sequence alignment as well as to study the relative comparable genes of interest or proteins of interest. In this bioinformatics recap, we saw more about how to access this protein data bank, which is also called as PDB. So generally, you type a four-letter code in this PDB database, and it will give you more information about how the protein is structured, what are the composition of its amino acids, and we also saw more about a software called String, or shortly called as String DB, which is used to study the interacting molecules with the protein. So if we type any protein ID of our interest, StringDB will give you the common proteins which are often interacting with the protein of our interest and what is the intensity of the interaction. And we also saw more about another software called Panther, also called as Panther DB. Panther DB more likely gives you information about gene ontology as well as your protein relationship through your evolutionary relationship. And after that, to study your to study and to visualize your receptor ligand interactions, we saw about a software called PyMol, and PyMol helps you basically to visualize your different proteins, and it also is used to study molecular docking. And we also saw more about another software called as PatchDoc, which is also used for molecular docking, and but in a more quantitative manner. It is used to study the strength of your docking sites. And in the last part of the lectures, last part of the course as well, we saw some facets about your ethics in research and publication, where we initially saw into reasons for scientific paper retraction. So the course instructor started by saying that there are a lot of papers in the today's scientific scenario where they are being retracted for retracted after reviewers find that there is wrongful information printed in those papers. And we also saw discussion about how an image can be manipulated. So here we have here I have pasted an example where we can see image manipulation, which can be used for your, which can be, which is the same image after manipulation can be used for a different study. And we also saw more about the different types of misconduct, where the three main types of research misconduct is fabrication of your data, where you fabricate your data points to align with your problem statement and your desirable data outcome. Falsification of your data, where something which doesn't work is proven, something which um, something which is known to not work is proven to proven to be worked by series of falsification of data. Like, you falsify the data, you change the date, you add, delete, or modify data to prove that something which is not working, which is working, or vice versa, something which is working is not working. And the third type of research misconduct is plagiarism, where plagiarism is basically copying of scientific data from any one of your journal-based data or your conference-based data or any other lab person's data into one's own data. Plagiarism is divided into like several classes, out of which scientific paper. It ranges from scientific paper misconduct, where you copy someone else's data, to self plagiarism, where you copy your own data in a different manner. So after this, we also saw some causes and cases for research misconduct, and how can we avoid this types of research misconduct. So that's all I have about the general recap of week 8. So I'll leave the room shortly for 5 minutes. So till we can have a short doubt session from 3 times 320 now. So we can have a short doubt session from 320 to 325. After which I can proceed to discuss the problem statements for week 8. If you have any doubts but you can't open your mic, feel free to type your doubts in the chat box in this area. Okay, if there are no doubts, we will move on to discuss about the problem statements for week 8. So, first question, which of the following statement best describes a fundamental aspect of systems biology? So, we also saw in the course recap as well as, sorry, we also saw in the week recap as well as for this TA session about this general systems biology. So, there are four options. So, option one, understanding specific genes and how they work within a biological system is the only focus of systems biology. Second option is the main method used by systems biology to simplify intricate biological processes is reductionism. And systems biology analyzes and comprehends the behavior of complete biological systems by integrating data from several biological organization levels. Fourth question is systems biology relies on isolated experiments and does not consider the dynamic interactions between components within a biological system. So, all these are correct in a way, but they are asking which is the following statement which best describes the fundamental aspect of systems biology. Does anyone want to attempt this question? It's a very theoretical question, so it should be easier to answer. And this kind of questions are most likely the ones you will get it in the exam. So the answer is simple. So system it analyzes and comprehends the behavior of complete biological systems by integrating data from several biological organizations. So it's a holistic approach, like I said in the week recap. So moving on, a student from IIT Bombay wants to perform quantitative proteomics with lab-grown cancerous cells. She has included SciLab, which is stable isotope labeling by amino acids in cell culture, in her experimental design. Which key aspect of SciLab will help her quantifying the protein abundance between different experimental conditions? Also, mention the potential applications of SciLab that would help her in a time course experiment. So SciRack is basically a technique which is used in which you label isotopes, certain isotopes of your protein samples using a label, and then depending on your label of your interest, it will detect your masses and it will quantify your protein abundance. So what is it going to be? So I've already given the hints. So it's going to label something and is going to detect the mass. So what technique it will be? So I'll go on to explain the answer. So the answer is it, since it is a mass spectrometry, so the aspect of SciLac which is going to quantify the protein abundance is relative peak areas in mass spectrometry. So it's going to use the relative peak areas to quantify the protein abundance between in different conditions. And the applications of SciLac is going to provide information on the protein turnover and protein expression. So it's going to give you information about your protein expression and protein turnover over time. So the correct option is option number D. So moving on. So question number 3, Parth identified a protein which plays a role in cancer therapy resistance. He hypothesized that the protein of interest interacts with genomic DNA while participating in DNA repair pathway. He designed the following experiment to validate his hypothesis. Identify the specialized experiment type from the following diagram. So this also I think we have uh, seen through the week recap. So I'll read the question again maybe. Parth identified a protein which plays a role in cancer therapy resistance. He hypothesized that the protein of interest interacts with genomic DNA while participating in DNA repair pathway. He designed the following experiment to validate his hypothesis. Identify the specialized experiment type from the following diagram. So if we look at the diagram, what is happening? So we have our gene of our interest and we have our protein which Parth identified, and that protein is somehow interacting with DNA and using DNAs which breaks down DNA. The DNA is broken into fragments, but the DNA which is interacting with the protein remains the same. So after that, what he is doing is there is Parth is administering an antibody which kind of binds to the DNA, and then the protein is kind of released, and then the purified DNA is made to interact with uh, synthesized DNA, custom made DNA, which is on a platform, and then it is used to amplify. So we saw this concept elsewhere in the recap as well as in the course as well. So what it is. So basically, what other technique will be used for your DNA protein interaction? So what techniques here? So by that as well, you can kind of remove options. So Sanger sequencing doesn't involve your proteins, so that can be removed. Multitoph involves proteins, but it doesn't involve your DNA, so that option can also be removed. So the remaining two uh, options are chromatin immunoprecipitation and reverse phase DNA protein microarray. Here chromatin just means the complex of DNA with protein. So I'll go on to discuss the answer. The answer is chromatin immunoprecipitation. So we saw about immunoprecipitation where DNA and protein interact, and then you isolate the DNA and protein of your interest by making it interact with your custom based antibody, and then you study the protein of your interest. So that is what basically is immunoprecipitation. So I'll go on to the next question. So this is a continuation of your third question. Is the above described experiment suitable to investigate whether the protein binds directly with DNA in vivo or not? Also mention if Parth can use the same approach, okay, can be used to detect protein-protein interactions in vitro. So this is a very simple answer. So your DNA, so this kind of immunoprecipitation techniques is used to study DNA protein interactions. So it can also be used to study if the DNA protein binds directly with the DNA in vivo. So the option is yes for the first question. But this same technique can be used to study protein-protein interactions. So the answer for the second question is no. So the correct option is option number one, yes, no. So question number five. So this I think you guys might have already seen in the previous questions as well. So the diagram describes a type of ionization source in mass spectrometer with the correct statement. So we just have to find what is the ionization source, whether it's a electrospray ionization, chemical ionization, MALDI, or your thermospray ionization. So here you can see that your green ones are your sample and the blue one is some other material, and the green one is kind of mixed with your sample is kind of mixed with the matrix, and once a laser hits, there is a desorption of your sample as well as your matrix, and it is generated into ions, which is then accelerated towards your mass analyzer. So what can it be? So we already know from the courses as well as the recap that is MALDI. So this is actually matrix assisted laser desorption ionization, where this the blue color represents your matrix and your green color represents your sample, and they are mixed together. So when the laser beam hits the matrix, it results in desorption of your matrix as well as your sample, leading to your generation of ions of a sample. So the correct option here is option number 3. The diagram describes the principle of Maldi, where the sample is uniformly mixed with a large quantity of matrix. I hope the explanation is clear. So I'll go on to the next question. So question number 6. From a rare type of plant, you have extracted a pure protein that you believe to be a fructose binding protein. Which experimental route will you choose to investigate the binding between fructose and protein? So I'll read the question once again. From a rare type of plant, you have extracted a pure protein that you believe to be a fructose binding protein. Which experimental route will you choose to investigate the binding between fructose and protein? So there are like four options here, and one is molecular docking, and one is performing a LCMS. LCMS stands for your liquid chromatography mass spectrometry, a combination of your liquid chromatography and your mass spectrometry. So basically here what happens is your sample is separated into its constituents using liquid chromatography, and the corresponding sample of your interest is passed through a mass spectrometry. And third one is studying the binding kinetics using SPR, which is surface plasmon resonance. Fourth option is both A and B are equally correct options. So does anyone want to attempt this question as to how can you study the binding between fructose and protein? So how this experiment, I'll also give a hint into how this experiment can be performed. So basically you will have a protein on a surface, and then your protein will give you a certain signal, and after your fructose binds to the protein, there's going to be a change in the signal. So through that you are going to study the binding kinetics. Okay, so anyways, since you are studying the binding kinetics, the technique is going to be SPR or surface plasmon resonance. So the correct answer is you will study the binding kinetics using surface plasmon resonance. So how it will generally happen is you will have a surface, a gold surface or something. There you will have different. There you will have the protein which fructose responds to. So you will have a incident light here and a reflected light here, and your plasma at a certain angle of incidence, your plasmons in the gold film start to excite and oscillate. So if you are tracking your uh, time, intensity over time, you are going to observe a baseline, and then you are going to observe an increase, and then sorry, it's good, this is going to be a baseline. So when your fructose binds to your protein of your interest at this point, it's going to cause a increase or a decrease. In some cases it can also result in a decrease. So it can cause a increase in the signal at the specific incidence angle, and once the fructose is washed off from the protein, the protein will also go off, leading to a decrease in your signal intensity. So this is how your surface plasmon resonance works. So does anyone have any doubts with this? So if not, I will go to the next question. So over time, the population of Indian women who are at risk for cervical cancer has increased. Towards an effort to raise awareness through scientific research, IIT Bombay has recruited a cohort of 100 women to be screened for cervical cancer risk factors. Which is the strategy would you like to follow? So IIT Bombay has not raised, sorry, recruited 100 women to study cervical cancer risk factors, and they are asking the question. Basically, asks for a strategy through which we can study the samples of all the 100 women for cervical cancer. So one of the options is protein microarray, one is multi-TOF for TOF, one is PCR, and one is BNC combined, that is multi-TOF combined with a PCR. Which one of them will be, which you think it will be useful for uh, to study cervical cancer risk factors for 100 women, not for like a audience. So the key or the hint here is the larger audience here, the larger uh, the larger size of the cohort. So I'll go on to explain the answer. So since it is like 100 women, you need you don't know what the sample volume that will be getting out of 100 women, and you also need a platform in which you can study all the 100 samples at once. It will be better if you can study samples of all the 100 women at once. So it's best to go with your protein microarray. So the answer will be you will screen the serum samples using protein microarray for autoantibodies. So in this way, you can take small volume of samples from 100 women, plate it on a microplate, a reader of a microarray, and then quote your antibody of your interest, which you think is a cervical cancer risk factor, and then check for their absorbance. And if the patients really do have a higher throughput or is responding to the protein, then the specific person is at a risk of having cervical cancer. Else it is not. If you guys don't have any doubts, I will go on to the next question. So, identify the protein and the origin of the protein with RCSF PDB ID 1 RC2. What is the organism and resolution of the crystal structure? Pick the correct option. So uh, they're asking us to yeah. So they're asking us to. They're given us a protein data bank ID, that is 1RC2, and they are asking what kind of organism is it and what is the resolution of the crystal structure. So you guys can try it now as well in your browser, or if you want me to try it out with you, I can also present my window and then try it along with you. But for now, what I have done is I have went to protein data bank, and in this box where it asks for a search term, I entered this ID. I'll tell you how to solve this for now. So if you type 1RC2, it gives you this page. You will land in this page where you can see the structure summary of the, where you can see biological assembly 1, and this is the structure summary, and then you can see the name. So 2.5 angstrom resolution, X-ray structure of aquaporin Z. So aquaporin Z is a protein, and they have done X-ray diffraction um, method, and then using this resolution they have got this structure. So this will be the page that you will be landing in. So they are asking what is the organism and resolution of the crystal structure. So if you go here, if you land in this page, you will find that the organism name here, Escherichia coli. So protein they have obviously asked is aquaporin Z. So protein will be aquaporin Z here. Organism will be E. coli or Escherichia coli, and your resolution is given here. Once again, yeah, resolution is given here, 2.5 angstroms. So your correct option will be protein aquaporin Z, organism E. coli, and resolution 2.5 angstroms. Is this clear to everyone? Uh, Ayman Sheikh, okay, yeah, you can ask it out.
So we can during the exam can we open the protein bank and check the answer and write?
So during the exam, I don't think you can exit the software, but I think they will give you image cues if they want you to find the protein data. If they give you a protein data bank, they, I think they will better give you interface where you can actually scroll only one page, and then you can, there you can find the details and then type it, or choose the correct option.
Okay, sir.
Yeah. But uh, I, so this is just for you people to familiarize yourself with this assignment questions or problem statements. Whatever questions you have also got in your week 8, this will also be for just yourselves to familiarize yourself with the software. And I don't think you will be able to access the browser in your actual exam. So the probability of them asking this kind of questions will also be very very less. And even if they ask, they will have some facility in the question paper itself, that where you can access the protein and then get the information. So for now, for solving the assignment questions, you just need to know how to access the data which is present in this page just by going into the protein data bank site. So I'll go on to the next question. I'll also have a separate doubt session after discussing all these problems. There you can also ask your doubts, or there also you can type your questions in the chat box, just like here. So next question is also a similar question. So identify the crystal structure for the protein fibromodulin from the diagram. What is the RCSB ID of the crystal structure? Pick the correct combination. So they have given the protein fibromodulin. So what you can do is you can go to the same protein data bank site. Here in the search term, you can enter fibromodulin, and if you enter the search, if you search if you type fibromodulin and enter search, this is what you will be getting. So this will be the first query that will be landing in once you search the fibromodulin. So if you can see here, the structure of that they given in this matches the first one here. So the structure of fibromodulin is 1, and they are given here the ID which is 5MX0, which is the RCSB ID. So the correct option is option 1, this for the structure, and the ID will be 5MX0. I'll repeat once again. So once you go into this protein data bank site, you can enter your in the search term, enter search term, search box. You can enter your fibromodulin, and then once you type fibromodulin, you will land into this query, which will give you the structure, 3D, the 2D structure, which will match with the number 1 query, and then it will also give you the main ID, which is 5MX0. And here they would have given your method of identification and the organisms. If they ask any other question, you can type that in the exam as well. And if you click into this 5MX0, it will land you in this kind of page where you can find additional details. So I'll go on to the last problem statement for week 8, after which we will be solving our assignment questions for week 7 and week 8. So Pranjal is a junior researcher at a cell culture lab, and he requires a specialized cell line for one of his experiments. Fortunately, many frozen vials of the identical cell line stock are in possession of a senior. However, the senior still won't give him the access to the cell vials despite repeated requests. Following many failed efforts, Pranjal was given one cell stock. Nevertheless, he was curious, so he conducted a STR, which is short tandem repeat profile, to verify the legitimacy of the specific cell line, and discovered that the senior had given him the incorrect cells. What action plan does he have? So this is just a theoretical value based question. So ideally, if he finds out that if he has been given a wrong cell line, he should try to contact a supervisor first, and he should ask the supervisor as to what can be done, since he has been uh, since he is suspecting a research ethics issue because he has been provided a wrong cell line. If at all the supervisor asks the senior about that and discovers that he accidentally given him a wrong cell line, then it will not be a research ethics issue. Otherwise, it will be a serious research ethics issue. So this is just a theoretical question which you can answer directly in the exam. So that's all I have for the problem statements for week 8. So time now is 3:51, so I'll probably have a we can probably have a doubt session till 4 or maybe. So after that I'll start discussing the assignment questions that you had got for week 7 and week 8. So the doubt session is still like four. We can have it if you guys don't have. We can start the assignment the discussion right away. But if you guys have doubts, you can open your mic and ask, or you can also type your doubts in the chat box so that we can solve it later. So Humaira is asking whether the exam, how the exam is gonna be. It is going to be only MCQs. So, generally, what happens is all the NPTEL based exams they are varied into like three different types of questions. So, one will be your MCQs, one will be your MSQs, and one will be your NATs. Yeah. So your MCQs are multiple choice questions where you will be given like different options and then you will be asked to choose any one of them. Your MSQs are multiple select questions where you will be given multiple options and you will be asked to choose all the correct options out of that one. So if you are given four options and multiple select, you can also choose two or three or four options. And numerical answer type questions will be NATs where you have to type your answer into numbers. They'll be giving a box like this chat box and you will have to type an answer. But for this course, like for all the problem statements that you have seen so far, I think there were very few MSQs, multiple select questions, and there were like no numerically answered questions so far. So, but anyways, be prepared for these kind of questions. So, these are all you will be asked to this thing. So, let's just say they ask a question like, what are all what? How many number of bases are there? So you know that there are like in DNA that how many number of bases are there in DNA. So you know that it's adenine, guanine, thymine, and cytosine. So there are four amino acids. So you have to type four in the box. So that's how it works. So you don't need to carry your calculator. So the software with which you are going to attempt the exam, it will have its own inbuilt calculator. So you can use that calculator. You have to use your mouse or keyboard to enter your values. So if there are no other doubts, if you have doubts as well, if you have further doubts as well, you can put it in the chat box. For now, we can move on from the doubt session and we can go on to discuss the assignment questions. So first we will start with the week 7 assignment questions. So pick the correct combination of question number 1 out of week 7. Pick the correct combination of statements. Okay. The questions in the exams are mostly from assignments questions. I can't say like that because I am not designing the question paper. Your course instructor is going to design the question paper probably. But some of the questions you can expect from your assignment questions, but not all of them. So pick the correct combination of statements that best defines the quaternary structure of a protein. So they have four statements. When two or more polypeptides join forces via disulfide connection, a quaternary complex is created. One of the statement number two is one of the amino acids required for the creation of quaternary structures is cysteine, and third is the quaternary structure is formed between peptide linkages where hydrogen bonds play a crucial role in the structure formation. Fourth one, aquaporin is not considered a quaternary structure, but hemoglobin is. So which one of them best defines the quaternary structure? So from here you can see that aquaporin is not considered as quaternary structure is false because aquaporin is considered a quaternary structure. And third one, quaternary structure is formed between peptide linkages where hydrogen bonds play a crucial role. This is also false because hydrogen bonds most likely form between your this thing uh, tertiary structures. What happens in the quaternary structure is it mainly forms disulfide bonds and other non-covalent interactions. So the correct options are 1 and 2. So 1 and 2 are correct. That's the correct answer. So any options which are having 3 and 4 are incorrect because 3 and 4 are not correct. So we'll move on to the next question. Which one of the following amino acids lacks optical activity? Assume that you are involved in the production of antimicrobial peptides. You wanted to measure the peptide amounts using the UV visible spectrophotometer after purifying them. Which of the following peptides will be the easiest to find at a monochromatic wavelength of 280 nanometer? Choose the appropriate set of responses. So, to solve this question, rather than going by the amino acids, I will rather encourage to go by the sequences that are present in the amino acids. That will probably lead you to the correct answer. So, one of the can is do someone want to solve this question first? Okay, if not, I'll solve the question. So, so like I said, instead of going with the amino acids, I think the easier way to solve this question is to look at the sequences. So, they are asking the second question, which of the following peptides will be the easiest to find at a monochromatic wavelength of 280 nanometer? So, in week 7's lectures, we saw that in 280 nanometer, the quantification of proteins can be done using the concentration of certain amino acids, tryptophan and tyrosine. So, tryptophan is represented by W and tyrosine is represented by Y. So in all these four amino acid sequences, in peptide sequences, which one of them have W and Y? Only the second one. So only in this sequence you can find the concentration of the peptides. So the correct answer is option number 2, fourth amino acid, as well as the sequence which starts at Y and ends with W. So I hope it's clear. So I'll go on to the next question. So this one, I think I have already discussed in the previous week's problem statements, but I'll explain it now as well. So you have purified a synthetic peptide that has 4 negatively and 8 positively charged amino acids at pH 5.6. You notice that when the peptides were treated with an alkaline solution at pH of 8, 4 positively charged groups were deprotonated. What is the current net charge of the peptide? What direction will the peptide move in a gel-based solution when the electrical field is introduced at pH 8? So I hope you get the question. So the peptide has 4 negatively charged amino acids and 8 positively charged amino acids at 5.6. And after that, they are treating it with alkaline solution of pH 8, and then 4 positively charged groups are deprotonated. So how many total positive charged groups are there? 8 minus 4, which is 4. So now the peptide has 4 negative and 4 positive charge. So the net charge of the peptide will be 0, because minus 4 plus 4 is equal to 0. And then the direction, it because it since the protein does not have any charge, the peptide will not move in a gel based solution when an electrical field is introduced. So the answer is option A. Net charge on peptide is 0. Movement of the protein will not happen. So next question. This also I think we have discussed in the previous problem statement, the previous week's TS session. So the image below shows the DIGE, that is differential gel electrophoresis method, which employs two types of tissue samples, normal and cancerous. And then Psi2, Psi3 and Psi5 were mixed with N, normal, cancerous, and a combination of both normal and cancerous respectively. Before applying the samples to the gel, they were mixed. After electrophoresis, the gel picture was examined for three distinct wavelengths according to cyanide dyes. Select the right answer. So your normal is stained with Psi2, cancer is stained with Psi3, and your pooled sample, that is normal plus cancerous, is stained with Psi5, and then is giving you some responses. And it is run with DIGE, and then you have to select the right answer. So the options are, the proteins in gel N plus C are entirely distinct. That cannot be true because your N and C will share some common genes, and some there will be some genes in N which will not be affected at all even though they are in cancerous condition. So this option will not be correct. First option. Second option is, the number of spots in gel is always smaller than both gels N and C. This also can't be true. Uh, it can be equal as well, or it can be less as well, but it can't be always smaller. So this is also incorrect. So if B is also incorrect, so both B and C are also incorrect. So this will be the correct answer. But let's just see. So the N plus C gel excludes the probability of batch to batch variation and is considered to be a good reference for further data analysis. So this is a right option, which we always do in DIGE. So the main advantage of using DIGE compared to your two-dimensional gel electrophoresis is to reduce the batch to batch variation, because you can image the gel from any dimension possible. So the answer is the N plus C gel excludes the probability of batch to batch variation, and it's considered to be a good reference for further data analysis. So if there are no doubts, I'll move to the next question. So fifth question. In a mass spectrometer, the ionization source performs which of the following function? Please select the appropriate ionization technique involved with the mentioned activity. Pick the correct answer from the following options. So they are asking which ionization technique will do which function. So soft ionization converts the unlight into gas phase ions in vacuum. Hard ionization is converts the unlight into liquid phase ions in vacuum. And soft ionization is in MALDI, the unlight is mixed with aromatic matrix and bombarded with of laser. Fourth option is in hard ionization, MALDI, the analyte is mixed with aliphatic matrix and bombarded with matrix. Is bombarded with a fast beam of argon or xenon atoms. This is called hard ionization. Which one do you think? This is also a pretty simple theoretical question. So does anyone want to try to give an answer? So it's pretty simple. The correct answer is option number 1 and 3. So the soft ionization, the unlight is converted into gas phase from vacuum. So that is soft ionization. And in soft ionization in MALDI, the laser is bombarding the matrix mixed with the analyte. So it is bombarded with laser. So does anyone have any doubts here in the first five questions, how they are being solved? We can have a like 2 minute break to see if there are any uh, confusions in the way that the first five questions are solved. Then we can proceed to go with the test. Okay, if there are no doubts till now, we can move on to the next question. So, sixth question. A volunteer gave his skin cells and blood cells for whole genome sequencing, and he found out that the genome sequence of both the cells is exactly the same. So, same volunteer, skin cells and blood cells, the genome is exactly the same. Help him understand why the two cells look and function so differently if the genetic material is exactly the same. So basically we are saying even though two cells have the same genome, why is the skin cells acting like the skin and the blood cells acting like blood cells? Like why are they behaving differently? So the answer is simple. So even though the genome is same for uh, both of them, the expression of coding genes in those cells differ, and therefore that results in the generation of different phenotypes. So answer is the expression of proteins in various type of cells is different at a different time. So I'll move on to the next question. In 2001, the first draft of the human genome was released. The objective was to read the entire human genome from start to finish. Scientists from all across the globe confronted a number of challenges in sequencing the whole genome, which was a phenomena does not describe the issues that have arisen in the Human Genome Project. So this is also a pretty uh, theoretical question. So the correct option is both A and C options are correct because your this was not the options A and C, that is sequencing the non-coding gene sites and sequencing the multiple replication start sites weren't the issue. But sequencing multi-gene sequence, especially in the shotgun approach, was a issue. So I'll move on to the next question. What is the PDB ID of the C-terminal domain of HPV16 E6 oncoprotein? What was the biochemical method of acquiring the 3D structure? So they given the name of the protein, that is the C-terminal domain of the HPV16 E6 oncoprotein. What was the biochemical they are asking the PDB ID and the biochemical method of acquiring the 3D structure. So this question you can solve it using, well, now that you have come in week 8, you can solve it using NCBA or using PDB. But for the sake of simplicity, I have approached PDB. So in PDB, this is what happens. So if you search this keyword, that is the, I'll take the pen. So if you search this keyword, C-terminal domain of the HPV16 E6 oncoprotein, this is what you get. So this is what C-terminal domain of HPV16 E6 oncoprotein. So this matches with this, and this is what you get. So here they have asked the protein data bank ID, which is this 2LJZ. So PDB ID is 2LJZ, which no nothing else has the same PDB ID. So this must be the correct answer. But still, we'll see the method of acquiring 3D structure. So the method here is, so they given method here, and then they given solution NMR. So here also, so solution NMR. So the correct answer will be PDB ID is 2LJZ. Method of acquiring 3D structure is solution NMR. So does anyone have any doubts with this? not, I can go on to the next question. So I'll go on to the next question. What is the length of the, so it's a continuation of the same thing. So you can click onto this 2LGSE page and then you can explore the other questions answers. So they are asking, what is the length of the C-terminal domain of the protein? Please mention it in amino acids. Also, write down the UniPro ID for the protein. It encodes. So if you click onto this 2LGZ button, then you will go to a page, and then if you scroll down, you will find this tab called macromolecules, where they have said sequence length is 75. So it has 70. So it means that the C domain, C terminal domain has 75 amino acids long. So the length is 75, and just below that they given the Unipro ID. There is P03126. Here also they given. Here also they given. So Unipro ID is P03126. So the correct answer is length is 75 and Unipro ID is P03126. Is everyone clear with this? Okay, then I'll go on to the next question, last question of week 7's assignments. What is the subcellular location of the above mentioned protein? Does it form any multimeric complexes? If yes, please mention and pick the correct combination of options. So in the same page, if you see that, you will find that it's a nuclear protein and it forms a homodimer in its active stage. So the answer is it is a nuclear protein. It forms a homodimer in its active stage. So that's all the answers for week 7. So this, I think, since the deadline is already over, you would have received the key. So I'll straight away jump into a short dose session till like 4:20 or something. Time now is 4:80. So till that you can ask any questions if you have in week 7, and after that we can go to discuss the week 8 assignment problems.
So for the question number 9, there was a um, um there was it yeah, it was they have asked to encode it and uniport ID and all. How are we gonna get to know? Because it's uniport ID and all, it's very difficult to remember and then
Same
select it
thing.
in the
And
exam, no?
that is the same thing. So just as I told you the problem statements for week 8, so I don't think so unless they give you a sheet or something. So it is possible that they will print out the first page and give it to you for like a reference or something. So, so you will find this in a web page, right? So they can print you the sheet and give it to you for reference. So unless that happens, you can't expect these kind of questions to come for the exam. So if this kind of questions come for the exam, you can expect a reference sheet to be provided to you where all the information will be given, and it will be your job to search the information in the sheet and then type it out. Select the answer in the exam.
Okay, so most of these questions will be from protein data bank only,
Yes,
sir.
yes. It will be like either you would have to access it from NCBI, or use like BLAST or cluster omega, or it will be from PDB, protein data bank, because in last week seven's uh, problem statements also we saw some problems from BLAST and etc.
So, do you think this exam will be difficult? Because most of the questions are related to this ID and all numericals.
Um, I don't think so. Like I said, it's not up to me to design the question paper for the exam. It's up to the course instructor. But still, I don't think there will be a lot of. Uh, you don't need to remember this kind of information for the exam. So for this kind of information, you will be given a reference sheet. Uh, and my suggestion will to remember the theoretical aspects of whatever you were taught from week 1 to week 6. So those will form the bigger chunk of questions for your main exam.
Okay, so thank you.
Yeah. So we'll finally just give me a minute. So we'll also discuss the week 8's assignment questions subsequently, and then we can have a common doubt session. Yeah. So question number one. So the number of Indian women prone to cervical cancer has leaped over the years. You wish to create awareness through scientific endeavors, and you have recruited a cohort of 50 females to screen for markers indicating the risk of cervical cancer. Which is the strategy you would like to follow? So this is a repetition of the same problem statement that we came across in the problem statements that I discussed for week 8. So if there are a lot of female patients, a lot of patient samples, and there are a lot of risk factors that you need to study about, which one will you be performing and which one will be much more easier? Which one requires a very small sample volume that you can use? It is protein microarray. So the answer will be you may screen the serum samples using protein microarray for autoantibodies. So if there are no doubts, I'll go on to the next question. Same. So the diagram describes a type of ionization source in a mass spectrometer. Pick the correct statement. So this also we discussed in the previous problem statement. So this illustrates the MALDI principle, which is the matrix associated laser disruption ionization. So the correct option will be option number D, which is the diagram illustrates the MALDI principle, which calls for uniformly mixing the sample with a substantial amount of matrix. So I'll go on to the next question, question number 3. So match the left column with the right column. So they are given three solution ionizations, and they are given 5 kinds of ionization techniques as well as, yeah. So this is the category and this is the type. So you have to match it with the right thing. So we already know that your solid phase ionization. So uh, ionization can be either classified as soft ionization or hard ionization, or this kind of gas phase solution or solid phase ionization. So we already know that your solid phase ionization, which is the C, it's going to be Maldi, because Maldi is a very organic molecule. You mix your sample with the organic molecule to form a paste kind of thing. After which the matrix will bombard it. So automatically your C4 will be the correct option here. If you find one, you can find the rest of the things. But your correct answer will be A2, B1, C4. But let's just see the other options as well. So your gas phase ionization will be your chemical ionization. Actually, wait. Yeah. So, yeah. Your gas phase ionization also can be your electrospray ionization. But if A1 is there, this the remaining two options are not correct. It is your solution phase ionization corresponding to fast atom bombardment and solid phase ionization corresponding to your photo ionization. That is not correct. So your only correct option remaining here is A2, B1, and C4. So the next question, in labeled mass spec instruments, the isobaric tag is composed of two different chemical groups which have specific properties. Select the correct combination of chemicals in the right order from the below mentioned diagram. So this is also a very theoretical question. So the answer will be your A will be your reporter group and B will be your balance group. So the balance group is just to ensure that the correct mass is obtained, and the reporter group is to just to provide the signature ion peaks in your mass spectrum. So fifth question, a student from IIT Bombay is performing a protein microarray experiment to investigate protein interactors. To detect two distinct types of samples against a printed protein, you have tagged two different colored probes with samples. As a result, you get a third color referred to the image below, which is definitely not a blank. Given only two types of samples to test interaction with, explain what is most likely the case. So, does anyone want to answer this question? Okay, so basically what they have done here is they have two distinct types of samples and then they have tagged them with two different colored probes. Let's just say sample one is yellow and sample one is green and sample one is red. So, uh, and then they are checking for the expression of the protein. If the protein is present in the sample one, then only you will get green color. Protein is present in sample two, you will get only red color. But in some cases, you are getting yellow color as well. So what does that mean? So it means that either your sample has mixed or blended, resulting in creation of new sample type, which through which your both the samples signal can come, or the protein is resulted in both the samples and overall there is no differential expression. So I'll move to the next question. So please select the protein microarray type from the below diagram in the correct manner. So this is an entirely theoretical question. So the correct answer will be option number 3, where one is reverse phase immunoassay, two is sandwich immunoassay, and three is the direct immunoassay. So in direct immunoassay, you have your analyte of interest. You produce a primary antibody which can detect the analyte, and then you provide a fluorophore-conjugated secondary antibody to detect the primary antibody. So this is your direct immunoassay. Second one is a sandwich immunoassay where you have your capture antibody first to capture the analyte, and then you have a detection antibody to capture the analyte, and the detection antibody will have a secondary fluorophore conjugated antibody. And this reverse phase immunoassay, you first have a capture antibody, and then you tag your sample with your fluorophore, and then you can try to capture it. This helps in more selectivity towards your sample. So coming back to next question, seventh question, DAPA, which is DNA array to protein array, was invented to overcome a few hurdles of conventional protein array technologies. Which of the following was the biggest hurdle of all? So this is a pretty theoretical question. So the answer is, so DAPA was mainly invented to, because they couldn't if you perform protein to protein microarray, you couldn't. There is a high. Biggest challenge was to identify and purify proteins to be used on the arrays. So what this DNA to protein array did was, it was be able to convert, provide a platform for the conversion of DNA to protein, which can be used for the microarrays. So the biggest hurdle was option number two, unavailability of purified functional proteins for immobilization on arrays. So eighth question, the protein LDHA, LDHA stands for lactate dehydrogenase, is one of the variables differentially expressed in the HPV, human papillomavirus positive and HPV negative human cell lines. Using bioinformatics, you want to identify LDHA's in silico protein interactors. What software or web server do you wish to employ as a result? Select the appropriate combinations from the list below. So, which ones will you choose? So, you can quickly search which one of the. So, you already know some protein protein interactors which to study protein protein interactions. So, PyMol is definitely a protein interaction software but is not used to study protein protein interactions. Rather, it is just used to visualize the protein interactions. So, this can be used. So, the correct answer will be options 2 and 3, which is your string, DB, and cytoscape. So, I'll go on to the next question. So, identify the crystal structure for 6W9C from the diagram. What is the resolution of the crystal structure? Pick the correct combination. So they have asked, they have given the crystal, three different crystal structures, and they have all given us the ID 6W9C, and they have asked us to pick the correct crystal structure, and they have asked us the resolution of the crystal structure. So as usual, so if you go to PDB and then type this 6W9C in the search bar, you will land in this page, and here it will be. So if you see the structure, it almost resembles the first structure rather than the second or the third structure. So the correct crystal structure will be 1, and for the resolution, you go to this experimental data snapshot where the resolution will be given as 2.7 angstroms. So 2.7 angstroms is here. So the correct option is crystal structure 1, and resolution is 2.7 angstroms. Angstroms is nothing but 10 power minus 10 meters. Is everyone okay with it, or can I go to the next question? Okay, last question. This is a pretty theoretical question. So the component of a mass spectrometer that separates ionized masses depending on charge to mass ratio is called a mass analyzer. Pick the one which is not used as a mass analyzer. So this is pretty simple. So magnetic sector, fast atom bombardment, quadrupole, and time of flight. And out of this, only fast atom bombardment is not a mass analyzer because it's a ionization technique. So fast atom bombardment is a process where atoms are bombarded from a quartz tube onto a sample in a very fast fashion to generate ions, so that the bombardment can happen. So fast atom bombardment is an ionization technique and not a mass analyzer. So the correct option is fast atom bombardment. So that's all I have for the doubt session. That's all I have for the discussion of the assignment answers. I can go back to the first to just give a recap of all these answers. So the first question, to screen the uh, 50 females for cervical cancer risk factors, you can use the serum samples using protein microarray. The option is B. Second question regarding your type of ionization source, it's option D, which is MALDI. So the third question, to match the left column with the right column, the correct answer is A. To gas phase ionization is chemical ionization. Solution phase ionization is electrospray ionization, and solid phase ionization is your MALDI. The fourth one, for the mass spec experiments, your A group will be your reporter group to provide signature ions for mass spec and mass spectrum, and the B group will be your balance group, which will have a constant mass. For the fifth question of microarray, the yellow color is generated because the protein has elicited responses in both samples, and overall there is no differential expression. In sixth question, the different types of protein microarray. The first one is the reverse phase immunoassay, second one is sandwich immunoassay, and third one is the direct immunoassay. So the correct option is option number 3, C. And the seventh question, the main hurdle that DAPA, that is DNA array to protein array, was overcame was to for the unavailability of purified functional proteins for immobilization on arrays. For the eighth question, if you want to use any software or web server to study the in silico protein interactors of LDHA, you can use String and Cytoscape. The correct option is option C, 2 and 3. So for the ninth question, to identify the correct crystal structure of 6W9C, you type the protein ID into this PDB, protein data bank, and there you get this crystal structure 1 as the correct crystal structure. And if you go to the resolution here, you can find that the resolution is 2.7 angstroms. And for the last question, the only technique here which is not a mass analyzer is fast atom bombardment. So once again, that's all for the discussion of the answers for week 8. So time now is 4:35 and the session ends at 5. So we can have the if anyone has any other doubts, you can ask, or we can. I thank you for coming to this week 8's TA session. And tomorrow I am going to take a extra TA session, which will be a general course recap, where I'll try to cover more problems from the all the 8 weeks, which will be useful for your exams. So that session is tomorrow, which is 16th March, Sunday, from 3 p.m. to 5 p.m. Same timings. So if you people have any doubt, I'll be in this room till then. But uh, uh, yeah, if you have any doubts, you can shoot them either through your mic or through the chat box, or I'll see you guys tomorrow from 3 p.m. to 5 p.m. Okay, if no one has any doubts, I'll see you guys in the next course recap session tomorrow from 3 to 5. Thank you.